webinar on applications of CRISPR Cas9 for disease research. My name is Tiffany Campolongo, and I'm a marketing manager at GenScript and the coordinator of the GenScript webinar series. I'm pleased to introduce today's presenter, Dr. Randall J. Platt, a postdoctoral fellow at the Broad Institute of MIT and Harvard. Randall works in the laboratory of Feng Zhang to develop and apply genome editing technologies toward uncovering the genetic and molecular basis of complex genetic disorders. His research establishes generalizable genome editing-based technologies and methodologies that facilitate the rapid testing of molecular hypotheses for causally linking genotype and phenotype in relevant developmental and physiological contexts in vivo. He obtained a PhD from MIT in biological engineering as an NSF fellow and an MPhil from Imperial College London in materials as a Whitaker fellow, and a bachelor's degree from the University of Utah in biomedical engineering and chemistry. Today, Dr. Platt will speak about his work on using CRISPR Cas9 genome editing technology to accelerate the creation of new in vivo and ex vivo models for disease research. After his presentation, there will be a Q&A session. If you have any questions during the webinar, please type them in the chat window. This webinar, including the Q&A, will be archived on the GenScript website. And now it is my great pleasure to turn it over to Dr. Platt. Welcome.
Hi, thank you very much. I want to thank GenScript for this opportunity for broadcasting this. It's a great honor to be able to speak to such a wide audience. And I'm speaking to you today from the Broad Institute, where I'm in Boston, where it is currently raining. So wherever you are, good morning, good afternoon, or good evening. So what I'm going to be talking about today is CRISPR Cas9 and the work we've been doing over the past several of years to engineer the genome to investigate disease mechanisms. And again, my name is Randall Platt. I'm a postdoctoral fellow within the lab of Feng Zhang at the Broad Institute of MIT, as well as Harvard. And so arguably one of the most exciting advancements of our time was the sequencing of the human genome. It not only brought into view the over 3.5 billion DNA bases of the blueprint of life, but it sparked new fields in biology and medicine where they were devoted to understanding what the sequence actually meant. So now, after decades of research, we're starting to understand what are genes, what are introns, what are exons, what are regulatory elements, and they're all quite fascinating. And also what the sequencing does is it enabled and sparked new advancements in genome sequencing technologies, namely next-generation sequencing. And through these efforts, we're not only sequencing single genomes, but we're starting to sequence many different genomes, and we can start to compare and contrast them. We can learn about interesting traits that make us different, and we can also learn about those genes, those changes, those mutations that are associated with any given human disorder. And that's where I think the ultimate power of genome editing and human genome sequencing lies. And so how this typical research model goes is it all begins with a patient. Here at the top, I'm showing you Vincent van Gogh, who undoubtedly had some form of neuropsychiatric disorder. And how we would investigate him in today's world is we would take people like him in the population as well as normal individuals, and we would sequence their genome, and we'd compare them, and we'd try to identify mutations that exist somewhere in the genome that cause risk for that given disease. And so for many different diseases, these lists can vary from maybe 5 to hundreds, if not thousands. And so these sequencing efforts give us a large list of genes that are associated with a given disease. So these are genotypes. And then it's our job as biomedical researchers to take this long list of genotypes and identify which ones are causal and what they are doing. And this is often done by engineering genotypes into model systems using genome editing tools. And once we have these genotype engineers in the model systems, we can start to investigate phenotypes that are caused by that genotype and see if they make sense in the context of human patients. And also we can use that model for investigating mechanism. And also we can use it for testing therapies that will one day make it back to the patient and hopefully make an advancement for their quality of life. And so there are significant challenges for each node of this circle I'm showing you. Each node and each connection between these nodes is severely complicated. But what I'm going to talk about today and what genome editing has to offer biomedical research is how we can speed up the pace and how we can go through a long list of genotypes engineer them into model systems, and investigate phenotypes to identify causal mutations. So what I'm going to talk about today is going from genotype to phenotype by engineering model systems. And this is what genome editing has to offer by engineering these genotypes into model systems, and that all relies on the fundamental process of DNA repair. If we can make targeted DNA double-strand breaks at specific locations, the endogenous cell machinery repairs it in one of two ways. The first way, which is shown on the left, is called non-homologous end joining. It is an act of desperation of the cell, whereby the cell can cause errors in order to insert the two ends back together, because if it does not, the cell will die. It will apoptose. And so through this act of desperation, it is random and causes errors. And if you create this DNA double-strand break in the coding exon of a gene, you can disrupt the coding frame and induce a premature stop codon, which will result in a knockout mutation. Alternatively, how the DNA double-strand break can be repaired is on the right arm shown above. It's called homology-directed repair. And in this case, the DNA double-strand break is the same, but now you're supplying you're supplying an exogenous template where you can switch out the genomic sequence for one you supply. And so this is a way of precisely modifying the genome, and these are called knock-in mutations. And you utilize this arm in order to induce disease-specific SNPs, small deletions or insertions, or even inserting fluorescent molecules or protein tags. There's a wide diversity of things you can do here. So again, through targeted DNA double-strand breaks at targeted locations, we can induce genome editing through non-homologous end joining or homology-directed repair. But as genome editors, we're only as good as our ability to create this DNA double-strand break. So over the years, a wide diversity of technologies have been developed for this purpose. Some of the earlier technologies were zinc fingers and tails. These are proteins, which are strung together and can recognize specific DNA sequences. And if these protein domains are fused to an effector complex or FOLK1 nuclease, they can induce double-strand breaks. But more recently, the CRISPR Cas9 system was discovered in nature and developed into a genome editing platform. And that's what I'm going to talk about today. So this image I'm showing you now is of a bacteria being infected by its form of a virus. It is a bacteriophage. And just how your body has developed an immune system to fight off these infections, so has this bacterial cell. And the name of this immune system that fights off the virus is called CRISPR. And in this complex diagram shown here is CRISPR and all of its subtypes. Building this system understanding this system as shown in this diagram took about 30 years, so a significant amount of work has gone into this at a life science and applied level. And it's a fascinating story, but basically how it works, its fundamental component is the bacterium is infected with a virus by the bacteriophage. The bacteriophage injects its DNA into the bacterial cell, and the bacteria is able to take a small piece of DNA, shown in red, and it is able to insert it into its own genome at a place called a CRISPR locus. And that small piece of DNA within the locus serves as a molecular memory of the infection of the bacteriophage. So at a later time, if that bacteria survives the first infection, it can recall that memory in the form of a guide RNA, a small piece of RNA called a guide RNA, which will guide in a silencing complex called Cas9 in order to cut additional invading elements. And there are many different systems for the interference portion, but all of them are utilized to destroy the invading genetic elements. And so genome editors realized that this could be a very useful system. It's an RNA-programmed nuclease that you can basically modularly target to different genomic sequences to degrade DNA. So what this system looks like now is just a two-piece system. There's the guide RNA, which targets Cas9, and the Cas9 nuclease itself. And it's easily to program to any different site within the genome just by identifying a 20 nucleotide sequence adjacent to something called a PAM, which stands for protospacer adjacent motif, which is any NGG trinucleotide, which occurs about every 8 bases in the mammalian genome. And together, this guide RNA can guide Cas9 to the site in the genome where it'll induce a DNA double-strand break, which again will go on to be repaired by one of the two repair pathways we talked about in the beginning. And what sets Cas9 apart from previous technologies is that this process is very efficient. Generally, you need to only design one guide RNA for any given gene. Also, because Cas9 is separate from guide RNA, for each new target, all we need to do is create a new and small piece of RNA, which we can do very efficiently. As with the previous technologies, we would have to build a new protein each time. Moreover, because Cas9 and guide RNA are separate, we can supply many different guide RNAs in order to target Cas9 to many different regions in the genome, which is useful for targeting different genes in different cells or many different genes in single cells, which has a wide range of applications. And the power of the system has been broadly recognized since 2013 and has resulted or been involved in thousands of different publications. It's been termed by Science Magazine as the CRISPR craze, because many people are using it for a wide range of applications. But in particular, what I was interested in doing is utilizing the technology to enable the editing of somatic cells directly in mouse tissue in an effort to overcome the need to build a traditional transgenic animal model every time. Because if Cas9 is so powerful and easy to target, why can't we just modify the genes in the cell types and the tissues we care about directly? Let's not build a traditional model. So what this system would look like is we have this piece of guide RNA in Cas9. Let's deliver it to the mouse tissues, let's say the lung or the brain, the tissue we care about, edit those genes, and study the consequence of that mutation. So trying to do this initially, it was very difficult because Cas9 is an incredibly large gene. It's an incredibly large protein. Its sequence is about 4.2 kb in length, and that's not counting promoter or regulatory elements. So it's difficult to deliver with classic delivery modalities. And so what we decided to do, since we're using mouse, why don't we just knock Cas9 into the mouse genome and overcome that delivery burden? So now all we need to do is deliver a small piece of RNA, which because of RNA interference, there's a huge platform that exists for delivering these small pieces of RNA. And since we were able to create this mouse and we're not limited by size, we could include all the bells and whistles of a transgene you may want, which are shown here. And we decided to knock the Cas9 gene into the ROSA 26 locus because it's known to be able to be expressed throughout development in pretty much all tissues. So this is the whole transgene in its full size, but the important elements are shown here. We have a CAG promoter, which is a strong promoter, constitutive and thought to be expressed in all cell types. We have Cas9, of course, and we also have EGFP to report that Cas9 is being expressed on those cells. And we also have this element called LSL, which stands for lock-stop locks, which essentially makes this transgene not be expressed until we also supply CRI. So the transgene is CRI-dependent. And given the difficulty of delivering Cas9 to tissue and expressing it, the first experiment we wanted to do is to see if we could induce Cas9 expression throughout the body. So we crossed the CRI-dependent Cas9 mouse to different CRI driver strains. Here on the top, I'm showing you tyrosine hydroxylase CRI cross, and on the bottom, I'm showing you a paravalbumin CRI cross. And so through these crosses, we can induce Cas9 in very precise cell types. And why this is important is because the brain is a very heterogeneous and complex tissue that is comprised of hundreds of different cell types. And oftentimes, we want to perturb genetic elements in some cell types, but not all cell types. And this strategy would enable you to do that. An alternative strategy is to cross the CRI-dependent Cas9 mouse to a ubiquitous CRI driver strain. We use the beta-actin CRI strain. And progenies of these crosses have all their organs glowing green, which is again is a reporter for Cas9, so it shows that we can express Cas9 throughout the body, throughout development, in most cell types. And at this point, this was great for us because we were able to overcome this expression boundary that we showed before, but this also got us to our next question. We're expressing a bacterial nuclease, a very potent and efficient nuclease throughout the body, throughout development. Are we causing any toxicity? Is Cas9 toxic in vivo? This is a very strong way to show that Cas9 may be limited eventually in biomedical research or maybe even as a human therapeutic. So this was a very interesting way to look at it. But it's also a very hard question to answer. How do you look at toxicity in an animal as a whole? We started with the broadest possible level, at the level of the whole mouse. Here I'm showing you on the left the CRI-dependent Cas9 mouse, in the middle the constitutive Cas9 mouse, and on the right a wild-type animal. And at the whole animal level, they are, for all intents and purposes, normal. They live to old age, they're fertile, they have normal progeny sizes, and they don't develop spontaneous tumors, and they don't seem to do anything out of the normal. But we wanted to go a little bit further, so we performed histological sections, did H&E staining to look at morphology of different tissues. We looked for cleave caspase-3, which is a sign of apoptosis, and gamma H2AX, which is a sign for DNA damage, and we were unable to find any differences in morphology, apoptosis, or DNA damage throughout these organs in Cas9 mice compared to wild type. But we also wanted to go a little bit further. We might not be inducing DNA damage or apoptosis, but are we changing the physiology of different cell types? And we answered this question by looking at a very sensitive cell type within the brain, which is a pyramidal neuron in the CA1 hippocampus. And we performed patch clamp electrophysiology with the help of Yang Zhu and Guoping Feng's lab at MIT. So if you look at the fundamental properties of these neurons, you can see that we did not change their action potentials. And if you look for even more fundamental properties like read-based current, input resistance, membrane capacitance, or resting membrane potential, we could not identify any physiological changes between wild type and Cas9 expressing neurons. And this was very exciting for us and a nice confirmation that we could indeed express Cas9, and we could do so without inducing any toxicity or physiological changes. But this also begs the question, are we expressing Cas9 high enough in order to achieve useful genome editing? And how we decided to answer that question is through making specific and spatial perturbations of genes directly in the mouse brain. So I built this AAV vector shown on the top right to express CRI to induce Cas9 expression, and also a guide RNA cassette to target Cas9 to a gene called NUN. This is an RNA splicing factor involved with Rolandic epilepsy, and we targeted this gene to a constitutive exon. And we packaged this virus into AAV serotype 1-2, and we injected directly into the brain, one side of the brain, into the medial prefrontal cortex. We only injected 1 microliter, waited 3 weeks, and this is what we observed. CRI shows the cells that were infected. NUN shows the target protein, and on the right, CRI-NUN shows a merge between the 2 channels. And what you can see is essentially all cells that were infected with the virus are no longer expressing the target protein. And to confirm that this was because we were actually cutting this target site, I dissected this region here, which was glowing green under the microscope, and prepared a library and performed next-generation sequencing. You can see that this target site here has small deletions and small assertions in the sequencing reads, which are telling us that this is the signature of Cas9-mediated editing. And to show that this was even more specific, we created a virus targeted to not NUN, but someplace not in the mouse genome, LAC-C, and we did not observe this knockdown. And to quantify this, we dissected out this target region, performed Western blots first. And on the left side, we have the NUN case, which is the injected side. We have CTR, which is the contralateral, or the opposite side of the brain. And we also have a wild-type animal not shown here. And what you can see is that only when you deliver the virus do you get Cas9 and CRI expression, and only when you deliver a virus do you get this robust knockdown of NUN. And if you perform replicate Western blots, you achieve about 80% knockdown of that protein. And if you, on the same sample, perform next-generation sequencing and indel analysis, you see that you find 80% of sequencing reads have indels. And the protein and the DNA match up nicely. So let me just blow up this fluorescence images to emphasize the point. In a matter of a couple of weeks, we were able to design this guide RNA, make a virus, and inject it into a region of the mouse brain and get a spatial and temporal knockout of a protein. And we did this without making a traditional transgenic animal model. And this was a very nice validation for us, but we didn't think this actually is the ultimate application of the technology, because using traditional techniques, we could make a single gene knockout or dependent mouse relatively quickly. But because Cas9 can be multiplexed to different areas of the genome, we can start to think about going from these monogenic models to polygenic models and do it rapidly. So we want to go from monogenic to polygenic. And we wanted to test this and model lung cancer. Lung cancer is one of the most common lung cancer types in the world. It results in about 1 million deaths a year, and therefore we understand its genetics quite well. And if you mine the literature, you can readily identify some of the top mutated genes, which are P53, LKB1, and KRAS. And so the idea here is, can we recreate these 3 mutations in a single mouse in order to model lung cancer, or more specifically, lung adenocarcinoma? But from a design perspective, it's important to know the character of these genes. P53 and LKB1 are tumor suppressor genes, so we only need to supply a single guide RNA to a constitutive exon in order to induce a frame shift knockout mutation. KRAS, on the other hand, is a proto-oncogene. We not only need to cut exon 1, but we need to induce a precise amino acid change. Here I'm showing you G12D, which should induce the proto-oncogene to promote tumorigenesis. And we wanted to do all three of these perturbations in single cells, so I designed a single vector shown here, which has 3 guide RNA expression cassettes, one targeted to each gene, R-loop to image the cells that are infected, CRE to induce Cas9, and this KRAS G12D HDR template, and that's to change the wild-type KRAS sequence for this gain-of-function mutation. So if you package this vector, we wanted to show that we could deliver it efficiently to the mouse lung. So through an intratracheal injection into the mouse lung, on the left saline and on the right the AAV9 virus, this is an intratracheal injection. This is essentially intubating the mouse and delivering a small amount of liquid into the lung. We showed that after 2 weeks we can get robust transgene expression, which was great. And we then waited about 2 months in the lungs of Cas9 mouse. This is what we observed. On the left are control lungs, animals injected with a guide RNA not targeted to these different genes. And on the right, animals injected with virus targeting KRAS, P53, and LKB1. And so after about 2 months, and sometimes even earlier, we see these large nodules here, which glow green under the microscope. And if you perform histological sections, this is what you observe. Again, on the left, LAC-C are what clear lungs look like, and on the right 2 panels are these KRAS, P53, and LKB1 injected lungs. What you can see are these very dense balls of cells, which are tumor masses, if you're not used to looking at these. And having a clinical pathologist investigate these, he does indeed identify these as lung adenocarcinomas. And so to emphasize the point here, it's not necessarily these genes that we care about. We've known about KRAS, P53, and LKB1 for a long time. We were able to induce these tumors, causing mutations in all 3 sites with high efficiency, with a simple design process, making virus and injecting and waiting 2 months. Whereas using traditional techniques where you need to modify each of these genes in an individual colony, interbreed the animals, that process could take 5 to 10 years. Now we can do it in a couple of weeks. So in conclusion, with the CRISPR Cas9 mouse, we've created this versatile platform for enabling the perturbation of genes in somatic tissue. In the presentation today, I showed you how we used AAV to modify neurons and cells within the lungs. But what I didn't have time to talk about today is how we used lentivirus in order to modify immune dendritic cells, which was subsequently used to perform a whole genome screen, and also how we use nanoparticles, a particle called 7C1, in order to deliver guide RNA to endothelial cells throughout the body of the Cas9 mouse. But if you refer to this Platt et al. Cell 2014 paper, you can see the particulars there. So in conclusion, we get to one of the most important slides of the entire talk, and that's the acknowledgements of all the fantastic people that made this happen. First of all, I'd like to thank my advisor, Feng Zhang, as well as many different individuals in the lab that make all of this happen. I also want to give special thanks to Phil Sharp and Sidi Chen for their help with creating lung adenocarcinoma within the Cas9 mouse. I'd like to thank Wuping Zhang and Yang Zhu for help with the electrophysiology of the Cas9 mouse. I'd also like to thank Robert Langer, Dan Anderson, as well as their students for helping us utilize 7C1 to edit the endothelial cells of the Cas9 mouse. In particular, I'd like to thank James Dahlman, and I'd also like to thank Aviv, Nir, Ramnik, as well as their students for enabling using lentiviruses to modify immune dendritic cells. I'd also like to acknowledge my personal support, my research support, and point out that these resources, both of the Cas9 mice and all the DNA plasmids used in this talk, are deposited and available to the broad academic community. Thank you for their time. Thank you for your attention. I hope you enjoyed the talk, and I'm happy to answer any questions at this time.
Okay. Thank you so much, Randy, for that wonderful presentation. While everyone is typing in questions to their chat box, I am going to review some online recordings and also some resources that GenScript has of interest. So I'm just going to grab presentership. Okay, so please keep typing in those questions, and I'll go through a few resources that you can use to help you in your GenScript, in your CRISPR research, sorry. Okay, so I'd like to briefly tell you about some additional CRISPR resources that we have. On the GenScript website, you'll be able to find our new CRISPR handbook. It's a free 36-page PDF download with a broad overview of the development of CRISPR biology and case studies showing how it's been applied for different uses across many research fields. We also have archived webinars, including one from just a few weeks ago by our in-house senior scientists on CRISPR genome editing in mammalian cell lines. And as I mentioned earlier, this webinar is being recorded, and it will be archived on our website in about a week. In addition, there are a couple of other recordings that are available online that may be of interest to you. So I'd like to highlight two conferences that took place in May this year. One is the Genome Engineering 3.0 Practical Workshop. It was organized by researchers at the Broad Institute, and it has recordings posted on YouTube by the MIT Alumni Association. And the other was organized by our partner, the Biopharma Research Council, and it focused on advancing drug discovery through genome engineering with an emphasis on stem cell engineering. And there are slides and recordings available through their website on this topic. And then finally, I'd like to mention that GenScript has in-licensed the CRISPR Cas9 technologies developed at the Broad Institute. So we can generate the tools that you need for your CRISPR research. GenScript offers custom gRNA plasmids, gRNA libraries, and genome editing services that deliver custom-made mammalian cell lines or microbial strains. And you can find more information about our CRISPR service portfolio on our website. Okay, so I want to thank everyone for attending and just remind you that when you exit the webinar, please fill out a survey that's going to pop up on your screen. It will be really helpful for us in terms of developing new webinars to deliver to you. And now we'll begin the Q&A session. So I am going to ask Randy some of the questions that you've sent in, and he'll give the answers. Okay. So one question, Randall says, they thank you first of all, and they say, how can we reduce the number of off-target sequences in Cas9 systems?
So how can we reduce the number of off-target sequences in Cas9 systems? So you must be referring to the fact, and many different papers that have referred to the off-target effects of CRISPR-Cas9. There has been many of these papers published, and it's a very complicated issue because the genome is a big place, and it's very difficult to assess as well as predict these events. So the question, how do we limit these? And I think the short answer here is we're still figuring that out, because right now we don't necessarily have a whole genome unbiased way to detect these. So even assessing whether we're reducing off-target effects rather than just shifting them, we're not entirely sure. But what we're doing now, and what many groups have done, have created bioinformatic tools that allow us to highlight guide RNAs with the least predicted off-target effects. So basically guide RNAs that are most unique. They don't have similar sequences in the genome. So that's the gold standard to date. Let's take these guide RNAs that don't have predicted sites within the genome.
Okay, thank you. Okay, another question says, I may have missed how long you expressed Cas9 in the mice, and if they have any disease pathology near the end of life.
That's a very interesting question. So these mice had Cas9 knocked in the genome, and we created two strains with this, one which is CRE-dependent and another which is constitutively active. So Cas9 is expressed from the single-cell stage until late in life. And when we performed those off-target analyses, I showed you those histological sections. Those were performed on animals that were very old. They were 6 months old, and so that's pretty old in a mouse lifetime. And we also looked a little bit later and we were unable to see any toxicity at those different ages.
Okay. All right. Thank you for that answer. The next question says, you just talked about 3 genes knocked out at one time. How many genes at most can you knock out at one time?
So I haven't tried more than 3 in my own hands. 3 was enough, and when you do that, oftentimes not all cells have all 3 mutations, so that is a limiting factor. But in the literature, people are starting to explore how many genes can you target, and the most recent one I can think of is a paper that came out of George Church's lab where they targeted, I believe, around 62 genes, and they were able to achieve levels that high. So what is the upper limit? We still don't know, but it seems like 62 is possible, which sounds like more than any one person could study at a time anyway.
Okay, thank you. Okay, the next question says, can Cas9 use non-guide RNAs, meaning other types of RNAs?
So that is a fascinating question and something we've thought a bit about and are still thinking about. And the answer is we don't know, but so far there has been no evidence to suggest that it is possible. We haven't tested that directly, but there are ways that you could envision doing it. If it was broadly a problem and it happened a lot, you would expect that eventually they would hit a gene that would cause lethality or cause cancer or cause some problem that would surface in the mouse, whether it be at old age. We've bred and shipped these animals. I've bred thousands of these mice, and we've actually shipped them to hundreds, if not 1,000 different labs, and we've never had reports of this happening. This doesn't say that it doesn't happen or won't happen eventually, but it has gone undetected so far.
Okay. The next question says, can you use the CRISPR system to knock in genes or maybe modify particular bits of genes, such as making point mutations, etc.?
Absolutely, and in fact, in the lung adenocarcinoma example I presented, we did just that. We created a precise gain-of-function mutation, and we not only changed one nucleotide, we changed, I think, 11 nucleotides in total, and you can do that using AAV, and you could potentially do that with other delivery vectors as well. But whether this is possible is going to largely depend on the type of cell type. It's thought that HDR, it relies on cell cycle, and so you might need to do this in cells that divide. Neurons, for example, do not divide, so if it does happen, it doesn't seem to be very efficient.
Okay. And you have a lot of thank yous for this presentation coming through. The next question says, can you please elaborate on how to design gRNA for point mutations? I've read from the Zhang website that PX335 vector with 2 guides flanking the target region is recommended. Can you comment on this? Thanks.
Okay, so there are many different ways to design a knock-in experiment, and it is largely going to depend on all of the different technical variables, such as, is this happening in cell culture, is this happening in lung cells and brain cells? And so there's really no blanket response for how do I do HDR in my system. That being said, there's a huge flexibility, and some are more efficient than others. If you're performing these experiments in vitro and cell lines, probably the gold standard is to utilize Cas9 to make the cut. 1 is generally enough, depending on the character of your mutation. So 1 guide RNA with Cas9 to create a double-strand break, and then you also supply the cells with a single-stranded donor template of about 200 nucleotides in length. And that seems to be the gold standard for just creating a SNP in cell lines. As we're in vivo, the space has not been explored that extensively, but it seems that some character of AAV induces or at least promotes the HDR response. So just the simple fact of supplying AAV to the cells can promote HDR. And so my go-to method for in vivo is to use AAV, and the efficiencies that we observed in the lung seem to support that.
Okay. This next question asks where the lung cancer project, the research, is published.
Where is it published?
Where is it published? Yes.
It's published in Cell. It was published in 2014.
Okay, I believe that was at the bottom of a footnote at the bottom of your slides.
Yeah.
And find that publication.
So if you go to the Zhang Lab website and go to publications, it should be in the list.
Okay. The next question says, how big of a piece of DNA can be inserted in a knock-in experiment?
I think this also is going to depend on the particular system and cell type, but in the literature it shows that you can create very large pieces. For example, large fluorescent proteins you may tag to a gene. For our experiment in vivo, we used an 800 nucleotide donor and just knocked in a single nucleotide change, for example. But you can go large, and I've even heard, I don't remember and cannot provide citations because I don't remember, but I think people have even done kilobase size insertions. So all of these are possible, but I think the size is largely going to be dependent on where you put it in the genome, what type of cell type. So again, it's very hard to give a blanket response, but I think we can go pretty big because we can even fuse chromosomes. I mean, there's a large diversity of things we can do.
Okay, so you have a lot more questions here. So I will ask you maybe 5 more questions, and if you want to keep going and keep answering them, that will be great, but I will leave that up to you. So the next question asks, what is the frequency of the homozygous mutants generated via CRISPR versus heterozygous?
So my favorite response to all these questions today has seemed like it largely depends. So in our study, we characterize the amount of the different types of mutations we see. How many are homozygous, how many are frame shifting, how many are heterozygous, and generally have to do this through single cell analysis because you want to know in the context of a single cell rather than in a bulk population. And we've done this for different targets, but in the case of NUN, it seemed like this particular guide RNA, this particular site within the genome liked to be repaired in precise patterns. For example, it loved single nucleotide deletions, and that's the reason that we saw so many knockouts. But if we do a different guide RNA to a different gene in a different context, we can see a hugely different pattern. So while some places in the genome have a distribution you would expect by random chance, but others seem to be more guided in their repair, and it might have to do with repetitive elements that are nearby. And so again, there's no generic response here, and if it really matters, then you would have to investigate it.
Okay. All right, the next question says, have you ever used a zebrafish animal model?
I certainly have not, but I believe there's extensive literature on that. But it's definitely been done in zebrafish. I just don't know the state of the field.
Okay. Okay, I'm scrolling through to find a couple more questions. Okay, this question says, how many SNPs can you replace in a single gene?
How many SNPs can you replace in a single gene? Well, you could imagine you could almost replace, so if you just replace an exon entirely, you could engineer many different SNPs. So for example, you don't have to do one SNP at a time. You can just replace a large chunk of a gene to replace many SNPs at one time. And so how many is limited by how big of a chunk you can swap out of the genome. And that gets back to the earlier question that we might be able to swap out hundreds, if not KBs. So I think the number of SNPs that we could engineer could go quite high as long as they're within a contiguous region that you could swap out with a donor. So in my case, for lung adenocarcinoma, we swapped in 11 point mutations, one that inferred the G12D switch, and others that basically labeled the HDR, which made our measurement of the rates of HDR much more faithful.
Okay. And so I have about 2 more questions here, and then I'll let you decide if you want to keep going. So the first one says, is the injection technology suitable for any organ? And what is the efficiency of CRISPR Cas9 introduction into particular organ cells of adult mouse?
So this is a good question, and different fields have developed different delivery modalities that will suit the given organ. The mouse as an entire system is a very complex place, and each organ, each system has a unique set of challenges that we may or may not have figured out already. So in the brain, AAV works fantastically. These cells take up the virus, and Cas9 is expressed robustly. But the lung, it seemed much less so. The lung cells didn't like AAV as much. Cas9 didn't seem to be expressed as high. And I think different organs and different systems within the mouse, you're going to find similar things. But you can start to change delivery modality, you can start to change promoters, and all these things I think you'll need to do. But to ask whether injection is suitable for different organs, I definitely have heard of it being done for many different organs. It's the most rudimentary way to deliver something to a given tissue. You just take a syringe and inject it. And I think this, with lack of better technology, is probably the way to start. But there's more sophisticated ways to deliver to places like endothelial cells, where we use nanoparticles, or the liver. You can just do tail vein injections with AAV or lentivirus, so on and so forth. So each system is going to have complex and unique challenges that may be amenable to delivery by different vectors that you would have to look into and probably have to design, try, and validate.
Okay, great. Okay, I have one additional question here. It asks, what was the most challenging experience in your CRISPR work?
Learning to work with mice, but CRISPR related.
Okay.
So to answer that question, what is the most difficult? I can tell you for the paper, the most challenging was making the Cas9 mouse to begin with, because as you can see throughout the paper, the experiments that we were doing were done on week time scale, but the process of making the mouse took a significant amount of time using traditional technologies. So sticking to the old system of making a traditional system took 1.5 years to maybe 2 to even build up the mouse in order to perform these experiments. So patience and tenacity to stick with your project was the most challenging for me.
Okay. So thank you for those answers. It's about 1:45, and so I will leave it up to you. Do you want to take a few more questions, or do you think this is a good place to stop?
How about I'll take 2 more.
Okay, 2 more questions. Okay. So one question says, how efficient is site-directed mutagenesis in changing multi-copy genes, for instance, greater than 30 copies per cell?
So that is a good question, and I don't have good evidence to support it, and I haven't seen anyone ask that question robustly in the literature. That doesn't mean it doesn't exist, but I certainly haven't seen it. And we know with increasing copy number becomes additionally challenging to do, because you have to do it twice rather than once, or 30 times rather than once, of course. And also with additional breaks within a cell, with additional double-strand breaks, you can start to see interesting things happen, especially if they're on different chromosomes, or even on the same chromosomes. You can start to see large deletions, you can start to see rearrangements, deletion, things like that. So a certain degree of genomic instability occurs when you have so many breaks within the genome. So I imagine achieving a level of 30 genes would be very complicated. So finding a way to break it down, making one happen after the others, sequentially or something like that might be necessary, because I would imagine 30 breaks within any given genome could be traumatic to a cell. But again, I'm not sure. I'm just speculating.
Okay. Okay, and I have one more question. It says, you just spoke about golden standard and HDR for SNP type of knock-ins. What are the golden standards from your perspective for repair templates delivered via double-stranded DNA or PCR products?
Oh, so gold standard in that regard is generally the bigger the better. I mean, the more homology you have to the genome, it seems to work more efficiently. So generally just a circularized plasmid is good because it tends not to insert randomly in the genome, whereas if you linearize a circular plasmid and put it in a cell, it could go in random places. So if the gold standard typically is the bigger the better, and double-stranded circular DNA.
Okay, thank you so much, Randy. I think that's all the questions that we have today, for today and all the time we have. So again, I thank you so much for joining us for this webinar. It was highly informative. And everyone, again, after you exit the webinar, if you could fill out that survey, that will be very helpful in us delivering more webinars like this to you. And again, I wish you great success in your research and have a great day.